Hi friends, today we are going to see the past year NEET questions for the chapter Strategies for Enhancement in Food Production. And today we are in the last part of this chapter. Today in this video we are going to cover part 6. Okay, so let's begin with the first question, which was asked in the year 2012. Consider the following statements from A to D and select the option which includes all the correct ones only. So you have to analyze the statements here. Single-cell spirulina can produce large quantities of food that is rich in protein, minerals and vitamins, etc. That is right. Body weight-wise, the microorganism Methylophilus may be able to produce several times more proteins than cows per day. Yeah, that's right. Common button mushrooms are very high rich source of vitamin C. Wrong. A rice variety that has been developed with a very rich in calcium. No, it was developed with rich vitamin A. Therefore, out of the 4 options, the first 2 statements are correct. Therefore, the answer is option D. Statement 1 and 2 are correct. Okay, next question here. Somoclonals are obtained by dash. I think you should know what the answer is, because when we talked about it in the earlier chapter, I told you that haploid cells are derived from the meristem region, while if you take the leaves, the roots or the buds, they're all diploid in nature. Now, what do you mean by somoclones? Maybe it's the time to discuss that. Or how can you describe somoclones? See, somoclones, or what is the meaning of somoclones? If you see, they are plants that are produced through the tissue culture and are genetically identical to the parent plant. Okay, so the clue is again here. The answer is option genetic engineering. The right answer to this question is, see, you have about 4 options here, right? You have about 4 options here. So I told you clearly it is by the artificial method that they are trying to do. They are not doing any natural method. So you have plant breeding, irradiation, genetic engineering and tissue culture. So somoclones, just now I said you are producing an artificial method where plant tissue cultures are used and that they will resemble their parent plant. Okay, therefore the answer is option D, tissue culture. Genetic variation present among plant cells during tissue culture is called as somoclonal variation. Maybe this part you need to remember. Somoclonal variation, that you have to remember. Okay, next question here. In order to obtain the virus-free plants through tissue culture method, the best method is... Yeah, this is a repeated question. If you had clearly watched part 5, this would have been easy for you. Yes, the right answer is meristem culture, because you have both the things there. Meristem culture is done by the development of virus-free plants. Meristem tissues can be taken either from the shoot or the root. Okay, fine. And this question was asked in the year 2006. Next question here. Cellular totipotency is demonstrated by dash. Okay, cellular totipotency. I told you what is totipotency, right? A single cell which has the capability or the ability to differentiate and develop and to form differentiated cells. So this cellular totipotency is found where? It is actually found in all plant cells. Cellular totipotency is exhibited only by plant cells, wherein plant cells retain the ability to deactivate and form new dividing cells. Therefore, the right answer to this question is option C. Next question here. In tissue culture medium, the embryos formed from pollen grains is due to dash. Again, it's an easy question for you. Clearly read the question. In tissue culture medium, the embryos formed from the pollen grains is due to dash. What do you think will be the answer here? It is because of the cellular totipotency. As we all know, it is the ability shown by many living things to form all types of tissue that constitute a mature organism. So sometimes they might be in deactivated form, but when conditions come, they might become activated and form different tissues which constitute a mature organism. Therefore, the right answer to this question is option A, cellular totipotency. So, totipotency is what I've given to your right side. In Latin, totis means entirely and potens means having power. The possibility of regenerating by entire plant from a single or few non-zoochotic cells was proposed by Gottlieb Haberland in the year 1854, 1945, and 1902. Okay, isolated cells from different differentiated tissue are generally non-dividing and quiescent. To express totipotency, undergo dedifferentiation and then redifferentiation. So now you understand what is totipotency. I've explained you in detail. The slide to your right side clearly explains that. Next question here. The reason why vegetatively reproducing crop plants are best suited for maintaining hybrid vigor is that. So what do you mean by, we all know what is hybrid, but what do you mean by hybrid vigor? Main question. See, it is the tendency of a crossbred individual to show qualities superior as those of, or I mean as the parents. That is, you're taking the sources from the parent, but the offspring actually exhibits character which are actually superior than that of parent. Very important here. They have characteristics that are more superior than the parent. Okay, so how can you actually say is best suited for post hybrid vigor? Is that option A is the right answer. Once a desired hybrid has been produced, there are few chances of losing it. Okay, so in vegetative reproducing, I mean a vegetative reproduction, the same genotype can be propagated from one generation to the next. Okay, next question here. Haploid plant tissue cultures are obtained from dash. What is the right answer? You don't have meristem here. Instead you have pollen grains, which is stands out because leaves, root tip and buds are diploid in nature. The answer to this question is pollen grains. All are diploid except pollen grain, which is haploid in nature. Haploid means N and diploid means 2N. Okay, fine. Haploid plants are preferred over diploids for the mutation study because in haploids dash. Okay, we have to analyze the statements here. Recessive mutation occurs expresses immediately. Induction of mutation is easier. Culturing is easier. Dominant mutation expresses immediately. The answer is recessive mutation expresses immediately. Mutations are generally recessive so that they are not expressed in their heterozygous condition. If a plant is haploid, then the mutation can express themselves. Therefore, the right answer is option A. Okay, next question here. Cellular totipotency was demonstrated by. So this is a direct question, not much of explanation is required. The answer is F.C. Stewart. He's and his co-workers in the 1964 raised the entire carrot plant from the carrot root culture. They established the cellular totipotency, that is, a capacity of a cell to develop a new plant from a single cell. However, the idea of totipotency was given by the German botanist Gottlieb Haberlandt, and we saw it in the last question where we saw a picture which demonstrated what is totipotency. Okay, but anyway, cellular totipotency was first demonstrated by F.C. Stewart and his co-workers in the year 1964. Next question here. In crop improvement program, haploids are important because they dash. Haploids are very important actually. Why? Why is it they require 1/2 of nutrients? Wrong. Are they helpful in the study of meiosis? Wrong. They grow better in adverse conditions? Wrong. They form a perfect homozygous. In crop improvement program, haploids are very important because they form a perfect homozygous, and therefore it is helpful to get a. So what is the ultimate requirement in the last? We need a pure line, right? And that capability comes from this haploids. Therefore, the right answer to this question is option D, to form perfect homozygous. Okay, so with this we complete the part 6 of this chapter. Hope to meet you in another new chapter in another video. Thank you for watching.